Okay, so we're going to talk about the various components and offerings of ISBCGC that allow end users to conduct cancer omics on the cloud. So just a brief outline. So I'll talk about what ISBCGC is, how users interact with our platform, and then what resources are available that can be used to interoperate with ISBCGC, and then end a little bit on the policy and security restrictions that users need to know. Manisha mentioned it already, and Bill talked about it this morning. ISBCGC is part of the NCI Cancer Research Data Commons ecosystem. Specifically, we are one of the 3 NCI cloud resources. The cloud resources are sort of the mission statement is to democratize access to NCI omics data to the cancer research community. So there's a lot of cancer data on the cloud. So we allow users to analyze that data, use that data. They can upload their own data. We provide infrastructure for large-scale analysis on commercial cloud platforms, and then more importantly, only authorized users, dbGaP authorized users who have access to the data can access the data. ISBCGC provides data as a service solutions to circumvent the common problems that users encounter when working with big data. So these are common problems not unique to cancer data as it continues to grow, and we provide a number of solutions that we'll go through in the upcoming slides. So just a little bit about our mission. So we provide NCI cancer data as well as high performance compute resources through the Google Cloud Platform in a couple of different ways. So first, we have an interactive web application for cohort building and data discovery, and easily accessible and queryable tables for multivariate data analysis. And we provide users with the capability to perform advanced pipeline and workflow execution on Google Cloud VMs. So our approach is we built an open platform for a broad range of users and use cases. We use existing systems to minimize development and maintenance costs. We leverage the best existing Google tools and technologies, collaborate with the research community on data standards and new data that they'd like to see on the cloud. And we provide a range of examples and tutorials to get people up and running on the cloud. So how do we make data available, or how do users access data on ISBCGC? So I just said we leverage and use a lot of the existing native Google technologies. So the raw data, either FASTQ files, BAM files, image files, that sort of thing, are stored in Google Cloud Storage buckets. And data that is tabular in nature, so clinical files, molecular files, gene expression data, mutation data, that sort of thing, and metadata manifests and stuff like that, are stored in a data structure in Google called BigQuery. So what is BigQuery, and how do we use it to enable omics analyses? It's a cloud-based web service from Google Cloud used to basically handle and analyze big data. So it's perfect then to use with omics data. It facilitates high throughput data analysis on the cloud and inexpensively in 2 ways. So users can store the results that are coming off of these large scale pipelines and workflows in centralized BigQuery tables. Google provides the first 10 gigs of storage free a month, and then 2 cents per gig thereafter. So users can store VCFs, MAFs, gene expression files, any kind of tab delimited file in a BigQuery table. And for analysis, it's really great because you can use standard SQL to query this large omics data. So the first terabyte of query data is free a month. This is a service that Google provides, $5 per terabyte of queries thereafter. Users can preview or interrogate the data without worrying about downloading data file by file, because the data is all in a single BigQuery table. And the nice thing about BigQuery is it seamlessly integrates with commonly used data analysis tools, like R, Jupyter Notebooks. Just a little bit more about the attributes of Google BigQuery. So it's a columnar database, so it's ideal for storing tabular data. You essentially get cluster computing power without having to manage a cluster. this is all done for you on the back end by Google. So users just really have to come up with really clever SQL commands to analyze their data. And because there's a lot of already hosted public data in BigQuery along with the data that we host, you can join tables to make and gain some really nice biological insights. So how do we leverage Google BigQuery? So for example, at the GDC, there are over 500,000 files, tab delimited files for TCGA data alone. So what we do is we combine data of a similar type into a single BigQuery table. So for example, let's say 150 MAF files of TCGA exist. We would combine those 150 MAF tables into a single MAF, or MAF files into a single MAF table. And then users could go in, write some SQL queries to query that MAF table now. Look for, build cohorts, look for associations and that sort of thing. So these aggregate tables can be queried rather cheaply and quickly on the Google Cloud. We have generated and we host over 300 open access BigQuery tables. And they're broadly in these three categories. There's derived data. So this is your gene expression data, mutation data, sort of the data that comes off of the workflows and pipelines. There's genomic reference tables. So these are the sort of the data that individuals use to make biological insights. So you would normally combine them with the derived data. And then we have metadata tables. These are sort of manifests or tables that are similar to manifests. So they include indexes of files, information about Google Cloud file paths or case barcodes and that sort of thing. So there are a couple of different ways of computing on data on ISBCGC. So we enable full command line access to analyze cloud-hosted data. So users have access to Google Cloud virtual machines and cloud-enabled notebooks that run both Jupyter and R. And using both of these mechanisms, they can interrogate, analyze BigQuery tables that they've generated themselves or that we host, and similarly can analyze or run pipelines or analyses on data that they have in their own Google Cloud storage buckets or GDC-owned Google Cloud storage buckets. So there are essentially 3 entry points into ISBCGC. And these are just some high-level examples of use cases. So we have a interactive web-based exploration. This is where users come in, they can build cohorts based on some clinical or molecular features. They can compare cohorts. You can upload your own private data set to analyze in conjunction with TCGA data. We have the direct command line access to VMs. Users can test, for example, new algorithms, programs that they've written on thousands of spam files, can run novel image segmentation methods on whole slide images. And then the interactive big data exploration and analysis. So this is, you can do interactive data exploration in BigQuery. You can access these BigQuery tables in R, Python to perform these multivariate analyses, and sometimes even develop and customize bioinformatics tools and pipelines. So after this, I'm just going to show a couple of screenshots of our web tools and each of the components that I'm showing here. So we provide a couple of different ways of data exploration through our web tools. So users can build cohorts using the web. Oh, is there a question? Oh, okay.
So I'll make sure we're muted if we're not presenting, please.
Okay. So yeah, so users can come into the web application, build cohorts based on clinical features or some molecular features. There are built-in interactive applications to view data. We have integrated visualization methods. So there's an integrated IGV viewer to look at read pileups, and we have 2 built-in image viewers to view histology images and radiology images. And they look something, so images would look something like this. And then as part of our collection of web tools, we have a BigQuery table search user interface. So I mentioned that we host over 300 BigQuery tables. So we have this user interface that allows users to explore and learn more about the tables that we make available. There are a variety of search features, so users can search by program or by category. So if you're looking for clinical data or manifest file metadata type, if you have a specific reference genome build you're interested in, or an experiment data of a particular experimental strategy, you can filter and look for data of that type. And what's really nice about the user interface, is once you've found and identified a table of interest, you click on it and there's a lot more details to really determine if this is a table that you're interested in. So each table has full descriptions about what the table contains, and there's even a bit of the schema as it shows kind of here in the picture. And then, so the benefits of this table search, we really like it a lot. It's no login required. So if you're just a browser, you want to know what we have, you don't need to log in. You can just come in, figure out if this is something that you want to use. We allow users to browse, learn more about the available tables. Each table, as I said, is really has been curated to include a lot of detailed information. So you can decide if this is something that this is a table that you're interested to work with. And another great thing is so you've identified a table of interest, you look at the field descriptions and stuff, it seems right, but maybe it doesn't contain the exact values that that you were looking for. We give you the ability to preview the first few lines of the table to see if this is actually the table you really want to be working with. So now once you've found a table you're really interested in, we have a really easy and seamless, basically you can jump from this table search UI to the Google Cloud Storage BigQuery console. We have a sort of this little red box shows a magnifying glass open button. You click on it and you get into the BigQuery UI. You can, you have access to that specific table. You can build your queries in there. And it's a very easy handoff essentially. As part of our web tools package, we also host the Middleman database, which is a manually curated, open access, highly used database of critical information for chromosomal aberrations and gene fusions. There's a user interface that users can come in and search for what they're looking for. We also have the same data in BigQuery tables, so users can also interrogate the data, not necessarily just in the UI, but also via BigQuery using SQL. So another avenue into exploring data on ISBCGC. So the Google Cloud VMs. This is a very busy slide, but essentially the take home is that users are unencumbered. They have unencumbered access to VMs on the cloud. So they can use VM resources as they need. They can scale up as they need. They can run any workflow language of their choice, or run any program or algorithm of their choice, develop algorithms as they choose, use any Docker images, etc. So there's a lot of freedom and flexibility to run whatever types of analyses you want. So, we've enabled, this has allowed us to sort of enable a lot of workflows and high level analyses for a lot of our, a lot of end users. And this is just an example of some projects. And then the final sort of entry point, which is Google BigQuery. So as I said, BigQuery, the nice thing about BigQuery is that it integrates with a lot of the commonly used analysis tools. So we're always using R, we're always using Jupyter Notebooks, and the ability to be able to call a BigQuery table from these interfaces is, I think, what really makes it attractive to data scientists and bioinformaticians, etc. So here's an example. So we have tables that contain TCGA data. We also have tables that contain GTEx data. So this is just a screenshot of the BigQuery console. Here at the top, we have the query editor with an SQL query. And what this is basically showing is using an SQL query to analyze the correlation between TCGA samples and GTEx tissue types. And you get the information back rather quickly. So in 18 seconds, it churned through 12 gigs of data. Again, tables can be joined in BigQuery using SQL. So you can draw some really nice connections. We have tables that contain genome reference information. You can join those with the derived data, to for example, look for pathways or gene ontology information. A typical workflow, users usually have 2 tabs open. So they'll have the BigQuery console open in the interface to build and experiment with queries. And once they've found a query that works with them and get the results that they're looking for, they move into their Jupyter Notebook environment or their R environment, write same query, push it into a data frame, and then generate some visualizations based on that data. Again, the nice thing about BigQuery is the ability to connect tables. So you can connect your own data. So if you have data you've generated in your lab, you can very easily create BigQuery tables. And then you can join them with ISBCGC hosted tables or other public data sets simply using SQL. So it's a very nice system. And just to emphasize again, the typical workflow or work setup. Users usually have the web interface, the Google Cloud Console open, where they're working on SQL queries, experimenting with the speed and time, how much data is returned, et cetera. And then they usually have a notebook of some sort open, and then always have some kind of like documentation page, maybe about SQL syntax or about, I don't know, how to pull in R's, BigQuery R or something like this. But essentially, this is the general work setup we'd imagine. Okay, so I said a lot, but what you need to know to interoperate with ISBCGC. So essentially, this is a very thin layer on top of the Google Cloud Platform. So users have full access to Google tools and technologies. They can run any type of workflow using any type of workflow language. They can come in with their own Google Cloud Project or AWS accounts. They have access to ISBCGC APIs and any and all Google APIs. Authentication and authorization occurs once using something called service accounts, which I'll talk about in a following slide. And what I think I've emphasized a lot about the BigQuery. You can store and compute on data in BigQuery. So BigQuery, you can save your metadata of manifests of GDC, which is what we've done, to find out where stored files are, so you can compute on them. We have compiled derived data in BigQuery, including reference tables that you could easily join to gain some really nice biological insights. We say this a lot to users who work on HPCs to kind of convert them to the cloud, but you don't need to wait on a queue. You have full pseudo access to your VMs, highly scalable in cores and RAM, use only what you need. Data backups are automatically managed easily, and you can easily manage access to your data by other groups. So just to talk a little bit about the security and permissions via service accounts. Service account is the ANA method that researchers compute runs under on the Google Cloud. Applications assume the identity of the service account to call Google API so that users aren't directly involved. So ISBCGC users will create a Google Cloud project, Google Cloud Platform project, and it automatically comes configured with a compute engine default service account. And then users register that service account with ISBCGC to get access to control data. These service accounts allow management of control data in terms of files, in terms of like directories, so things stored in cloud storage, for example, and data structures. So you can lock down BigQuery tables, for example, so that only users, authorized users have access to certain BigQuery tables. Researchers with validated service account use all the Google Cloud's resources natively and seamlessly. And just one more plug for Google Cloud. So Google Cloud Platform has a free tier, and it lets you compute without entering a credit card. So in the middle here, it's just showing a bunch of different things that you have available to you as a Google Cloud free tier participant, I guess. But the nice thing is, again, for 1 terabyte, you can do 1 terabyte queries per month for free. And then you get a rather small but efficient compute engine that you can compute on. I think I'll just end with this, but some example typical use cases. So we have users that fire up virtual machines to run pipelines using any flow of their choice. Another common use case is you want to build cohorts on the web application, and you can download file manifests of your cohort with locations of files to use for analyses. But the ISBCGC, I'd say gold standard use case, and we'll feature this in our demo tomorrow, is doing analysis using BigQuery. So use BigQuery to identify useful public data. Usually users start from our BigQuery table search UI. You identify a table that you're interested in. You want to interrogate it more. You want to look at the data. And then you move into the BigQuery console. From there, you can write a bunch of just SQL commands to look and see the kind of results you get. And from there, you transition into some kind of notebook environment, R or Jupyter, to perform some kind of multivariate analysis. Once you're in that environment, you can bring in any of the R packages. For example, bioconductor. There's a lot of bioconductor TCGA specific packages, and you can run them on our TCGA, BigQuery tables. And you can also combine your data with public data rather seamlessly. And then at the end, and this is sort of the bread and butter, right, of what most bioinformaticians want. You want to generate some really nice figures that your PI would love, and you can do that really, really nicely. So we'll go over that tomorrow in our demo. But I think that we end with that. Any questions?
We have plenty of time for questions. Please feel free.
So I'm curious, this is Alex, can you hear me all right?
Yeah.
Okay. I'm curious, like, what kinds of things have you seen users do to sort of mash up different parts of Google with what you provide?
In terms of like, are they using like a Google API plus our BigQuery tables, for example, something like that?
Yeah, exactly. Like what kind of technologies are they combining together? Have you seen some really interesting stuff from what they're doing, for example?
Actually, an end user just sent an email about something like, there's a lot of native BigQuery APIs. So they've used they figured out how to use the native BigQuery APIs to pull in pull in data from one of our BigQuery tables, and then they run their analysis in an R notebook that's cloud enabled in the AI platform. So basically they're not going into our web application to identify the table that they want, but they're using sort of the Google native API to pull in the data. So I've seen that sort of thing. They also can do the analysis in a Google VM. We've seen that where they run sort of BigQuery type analyses on an RStudio instance that they install on a virtual machine, and then pull in our BigQuery tables and do the analysis there.
We've seen some AI and machine learning as well, and you can even use that with BigQuery as well. So some of our end users are starting to explore that.
Okay, cool. Thank you.
Hi, this is Brian from OHSU. Just a quick question. How do you deal with harmonization of the metadata between the different projects? Do you address that at all, or is that left up as a user exercise?
What do you mean by the different projects? Do you mean like Target versus TCGA versus? Yeah,
exactly. Or if we look at the variety of projects involved in the Anvil, individuals, samples, specimens, etc., all have different attributes per se, and one of the goals is to allow people to query and do analysis across projects.
Yeah, so we have BigQuery tables of, so most of our data comes from the GDC. So we have all of the projects that are associated with the GDC, and all of the metadata associated with those projects that we pull from the GDC API. And then we create tables of that metadata. So patient information and the files that belong to that patient, like, and the sample information for that particular patient. So it would be like case barcode, which is the patient identifier, the samples that belong to that particular patient, aliquot barcode IDs, the files for which that basically like that were generated from different assays that were run on that particular patient. And all of this is in a series of like metadata tables that we generate every time there's a new data release from the GDC. And users can then come in and, for example, if you wanted to identify all of the RNA-seq data for a class of patients that have kidney cancer, you could run this kind of query on this kind of table to pull out disease code and then get file paths and, you know, for those particular patients. Does that answer your question?
Yeah, just one little follow-up. So when you say you're getting the data from the GDC, and the GDC has done a fairly extensive level of harmonization within their projects, does that represent the sum total of the projects you have on the platform, the GDC projects?
Yes, so we're actively working, so we're keeping up with the data releases, but yes.
Yeah, understood. Thanks very much.
Welcome.
Answered the questions. Yep.
Any additional questions? All right, well, please join me in thanking our speakers today. They had the monumental task of whittling down 8 hours of material down to 3.5 to 4 hours. So thank you all so much.
Okay, thank you.
Thanks, everyone.
It was really fantastic. As a reminder, tomorrow we'll be covering the use case portion of the training session. We'll start at 11 o'clock again on the same Zoom channel. And another reminder that we have spots open for the virtual Q&A with the speakers. These are short 10-minute sessions with the goal of really just getting us to interact with one another since we're all trapped in our homes. Thank you again to the presenters and everybody for joining.
Sorry,
Thanks, Mo.
make our own room?
Yes, your own room, or I can make one for you if you don't have access.
Okay.
Ping me.
I'm not sure I do. Thanks. Thank you.
Thanks.
Thanks,
Thank you.
everyone.
Thank you, Mo, for leading this effort through its twists and turns. You've done a great job.
Thanks to the audience. All right. We'll see you all in the virtual Q&A. Happy St. Patrick's Day.
All right, thanks everybody.
Don't go out drinking, anyone.
Yeah, all right.
Thank you.
Have one at home.
Thank you.
Join us on the Q&As and bring one with you.